Hi everyone, welcome to the fourth and last lecture of the biochemistry seminar for Lumiere. Today we are going to talk about mechanisms of CRISPR-Cas systems and their applications. So I want you guys to remember the DNA damage response and how DNA damage is repaired. So this is important because CRISPR, in essence, uses the host cells' DNA damage repair mechanisms in order to complete the edit. And so there is a crucial thing with CRISPR is that we want, when the DNA is damaged by the CRISPR machinery, and it's damaged on purpose so that way the edit occurs, but when it's repaired, we want it to repair via HR, homologous recombination, because remember, this pathway is error-free relatively, and it is more accurate, versus NEGJ, non-homologous end joining, is error-prone. And when we're making a genetic edit, we don't want there to be errors in the edit. And so we would want for the DNA to repair in the more precise manner. But how does CRISPR-Cas9 work? So basically, CRISPR-Cas9 works through this mechanism here. So you have your target DNA, and this would be like the chromosome, and you have this guide RNA, and the guide RNA is guiding the Cas endonuclease to the target DNA site, and the Cas endonuclease then cuts the target DNA in the area that this guide RNA is telling it to. And this is what elicits the editing to occur. So a cut happens, and then basically what happens is repair mechanisms occur. And so if it repairs by NAGJ, you could get an insertion or deletion or an indel. And that's not good because you want it to be precise to what you planned it to be. But if you give it something like donor DNA, and this donor DNA contains the correct sequence, it will elect to repair by homology-directed repair or homologous recombination. Remember, those are the same thing. And this results in precise editing. Now, there's different kinds of CRISPR-Cas9s. So there's a Cas9 endonuclease called dCas9. And basically what that means is it's a catalytically dead CRISPR endonuclease. So it's not going to make the cut that regular Cas9 does. So why would you want this? Well, because the Cas9 endonuclease follows the guide RNA, we can fuse other proteins to the dCas9, so that way it brings these other proteins to a targeted location through the guide RNA. And so there are different kinds of CRISPR experiments you can do with this, depending on what protein you fuse to the dCas9. So for CRISPR-A, or CRISPR activation, you can fuse a protein like VP64 to it, and this activates transcription in whatever region that dCas9 is being targeted to. There's also CRISPR-I, or CRISPR inhibition, and here you could fuse a Crab protein to the dCas9, and this would result in repression of transcription. So if you don't want a specific gene transcribed, you can target the dCas9 to go to that gene, and then the Crab will repress it. You can also do base editing, and so you could fuse a protein called ApoBec to dCas9, and this would result in a single nucleotide base edit. And so if you were only looking to change one nucleotide, then this would be the perfect option for you. Now, CRISPR-Cas9, while it's the most popular, is not the only CRISPR. So, another kind of CRISPR is CRISPR-Cas12. And CRISPR-Cas12 is different because it doesn't use a guide RNA. It uses something called crRNA, which is just a little bit different in its structure to guide RNA. But it does pretty much the same thing. It's just a different version of CRISPR-Cas9. It also needs the PAM site, which is the protospacer adjacent motif. And this is located at the 3' end of the spacer sequence, and it consists of a single A, U, or C base pair. And this basically allows the guide RNA or the crRNA, the CRISPR RNA, to find out, to know that it's in the right area. And, I was mistaken. So the PAM is not a single nucleotide. This, which I will explain, is a single nucleotide. It works similar to a PAM. PAM is a couple base pairs, 3 to 5, helps orient the Cas endonuclease to know that it's in the right region. There's also CRISPR-Cas13. So CRISPR-Cas13 is different because it edits RNA. And so you can edit mRNA that's going to be translated into protein so you can edit it post-transcriptionally. And this contains the PFC, which is the protospacer flanking sequence, and this is similar to the PAM, but it's only one nucleotide. Now, there are a lot of cool molecular biology techniques we can use with the CRISPR-Cas system. So, one thing we can do is we could put a guide RNA together with the dCas9 that's got a tag on it, and let's say this tag is a biotin tag. So then you transform both of these into cells, and that results in vivo biotinylation. And that will biotinylate the DNA at whatever location that this guide RNA that you gave it, that you programmed it for, to it. And this allows for us to do things like proteomics, RNA sequencing, and DNA sequencing, and visualization of where certain proteins are, like this tagged protein, that, like, where they go to when they are at the locus of the genome that it's targeted to. Oops. Another thing we can do is we could tag it with GFP, and that allows us to actually learn more about Cas9 itself. So when you tag it with GFP, green fluorescent protein, it allows us to see the Cas protein as it is working on the DNA. And so this allows us to learn more about how CRISPR works. So how can CRISPR be used in molecular biology and in medicine? So in molecular biology, CRISPR can be used for many different things, such as knockout or knock-in mutations. It can create libraries, it can target RNA, and you can actually see interaction of Cas9 with other proteins. the knockout knock-in is like if there's an oncogene, which is a gene that will result in, or it pushes to move into a cancerous state, we could knock out those genes versus tumor suppressor genes. Say you have a defective one, we could knock in a correct tumor suppressor gene. We could also knock in chemotherapy resistant genes to help relieve some of the bad symptoms that chemotherapy elicits. We could knock in metabolism related genes to help people's metabolisms. We can create CRISPR libraries, and this is where basically you use the dCas9 and you sequence the area that is, so basically there's like a little break, and the dCas9 comes in and fills in, because the dCas9 has something attached to it. And so like if you're doing activation, whatever's attached to the Cas9 will fill in this gap and result in activation, versus if you're doing interference, it's the same thing except it results in suppression of the gene because you have a different protein that's tagged to this dCas9, like I explained before. In medicine, so CRISPR can be used for diagnostics, for combating carcinogenic viruses, for animal modeling, and for immunology therapies. So like targeting T cells to basically programming T cells, which are a type of immune cell, to recognize an antigen on a tumor cell. Or the same thing can be done here as well. This is just a different mechanism by how it can work. You can also combat carcinogenic viruses by editing the virus's DNA itself and making it unrenderable. We can also use it in animal models so we can understand how using CRISPR in an organism is affected versus just looking at it in a single cell. And we can use it for diagnostics as well, because it's a very good search and finder, basically. And so that's what it's doing in a diagnostics context. So now you should be familiar with what CRISPR-Cas9 is, what dCas9 is, different types of CRISPRs, and different ways we can use CRISPR in molecular biology to help us learn more about CRISPR, but also to help treat diseases like cancer. If you want to learn more about CRISPR, I suggest reading this paper. This is a very good review that goes over the different kinds of CRISPRs, how CRISPR-Cas immunity can be inferred, and how DNA repair and genome stability is affected by CRISPR-Cas. Okay, thank you all.